Good afternoon, everyone. Thank you for joining us for this episode of the GHGA Webinar series, exploring challenges and benefits of metagenomics. I'm delighted to introduce today's speaker, Oleksandra Karpiuk. She's the project manager for the sequencing facility at the NGS Competence Center, Tübingen, located at Tübingen University Hospital. Oleksandra and I were actually from the same PhD program. That's where we actually initially met, and that's where she got her PhD in molecular biology from Göttingen University in 2013. Following her PhD, she gained extensive research experience as a postdoctoral fellow first at Max Planck Institute for Immunology and Epigenetics in Freiburg, and also later at Klinikum Rechts der Isar in Munich. In 2021, she moved to Tübingen to work as a staff scientist at the Max Planck Institute for Developmental Biology, and her journey led her to the NGS Competence Center Tübingen in 2022, where she now plays a crucial role as a project manager, overseeing various sequencing projects, ensuring the smooth operation of the facility, and providing this invaluable support to researchers. In today's webinar, she will guide us through the fundamentals of metagenomics and metatranscriptomics, also highlighting the critical considerations ranging from project planning to analysis techniques. And she will also discuss key applications of these methodologies, including their use in human gut microbiota, environmental biodiversity, and also disease diagnostics. Oleksandra, we are excited to have you share your insights and experience with us today. Welcome.
Thank you, Koray. Thank you for the nice introduction. That's a pleasure, actually, to be introduced by you. Let me share my screen. I hope that's going to work. Yes. So yes, the introduction was really great, so I don't have to tell you where I come from and what facility do I work in. So let me then jump directly into the topic. So our facility is mainly associated with the clinic in Tübingen. However, we also work with other universities, not only in Tübingen, but all over Germany and even with some other European countries. And our sequencing facility, which is called NCCT, is a part of a German-wide sequencing excellence network that is actually including several big sequencing centers, this network was started by the DFK in 2019, and the idea was to bring together facilities with the state-of-the-art equipment and workflows, and to help scientists with large and very expensive sequencing projects to get good quality of data out of their projects. The network was funded until last year, and from this year on, we're an independent sequencing facility network because working together worked really well. And we are also now collaborating with DFK with large-scale sequencing projects. So we'll be really happy to help you with the planning as well as the grant proposal if you have in mind a very interesting research idea. And with that, let me jump to the topic of the metagenomics. And before we go into details, let me firstly quickly tell you what metagenomics is. So nowadays we define it as the branch of genomic research fields that study collective DNA or RNA from the sample. So that means that all the organisms present in the sample will be investigated and the data from them will be obtained. The main breakthrough of metagenomics in comparison to traditional studies is that there is no need of pre-culturing or any way of expanding the species before actually going and sequencing them, because that would introduce bias, of course, and then the data that we get is not necessarily mirroring the real data that is actually in the sample. The metagenomics, as the word and as the term, appeared in 1998, and that was, at that time, it was mainly referred to the studying of soil genomes because it was so difficult to actually decipher the microorganisms of soil. However, the basic metagenomic techniques were discovered much earlier, and one of the pioneers in metagenomics is Carl Woese, who also is known for discovering archaea as a branch, and he actually developed methods of taxonomic profiling that we're still using nowadays and that are still very popular and very established. So why would one choose doing metagenomic sequencing? This method has many advantages. First of all, it is unbiased and it's hypothesis-free. That means that you do not have to know which organism you're interested in. You collect the data from all of them, and then you can choose what is the most exciting. It also gives you comprehensive data that you can come back to if you're interested in, for example, other species. Moreover, metagenomics is one of the not many sequencing methods that allow us to obtain novel information, meaning that new species can be discovered, new genes or gene clusters, and that's really difficult to do any other way. And finally, the turnaround from sample to meaningful data, meaningful bioinformatic data, is usually much faster than if you will go through traditional methods and isolate every species and sequence them separately. Now, when we speak about metagenomics as a whole, there is a lot of confusion about what exactly it is and which methods are included in it. So nowadays, we actually unite several workflows, lab workflows, and bioinformatic workflows under this word. And the main ones are shown here on the slide. So it's the amplicon sequencing, which basically means sequencing of a certain part of genome of many organisms. I will speak about that a bit later. We also call that targeted metagenomics. Another part that most probably you heard is the shotgun metagenomics. I will also explain how that works. And nowadays, we also include metatranscriptomics under the term metagenomics. And basically, it's really similar to the DNA-based methods. However, this method is mainly focusing on RNA. All right, so let me jump in and introduce you these three main branches of metagenomics and their similarities and differences. So I start with the amplicon-seq, and here it's the method that is using DNA as a starting material. And as I already mentioned before, you do not sequence DNA of basically all DNA of the species, but you sequence a certain gene cluster. You might have heard about this method if you heard about. So basically, after that, this certain gene or gene cluster is amplified, and it is sequenced and analyzed after that. And as I mentioned before, this we call targeted sequencing. Unlike amplicon sequencing, the shotgun metagenomics is untargeted. And in this case, the DNA of the sample is usually fragmented, barcoded, and then everything is sent to the sequencer. So basically, we cover all the species and all the DNA present in the sample. And metatranscriptomics is very similar methods, but however, we start with RNA instead of DNA. So we have to go through the RNA fragmentation and usually conversion to the cDNA, and then this cDNA is sequenced further. And this metatranscriptomics is also an untargeted sequencing approach. So however, these approaches are quite different. They all go through the same stages when you plan a metagenomic project, and these stages are listed here. So you, of course, start with material collection, your sample collection. Then one has to isolate nucleic acids from the sample. Then they go through the library prep and sequencing, and at the end, the data is analyzed. So usually at least in case of our facility, usually we are not responsible for material collection, and we can assist with the nucleic acid isolation if needed. However, these are mainly common protocols. But starting from the DNA or RNA that you obtain, after that, it's usually taken over by a sequencing facility, and all the sequencing-related preparations like library prep and sequencing itself are also done at the facility. We also do partially data analysis, so initial analysis like demultiplexing your data and also quality trimming. And we also can assist with primary and secondary bioinformatics. However, usually the scientists know better what exactly they want to get from the data. So now I will go through each of these steps and we'll try to explain you what is the most important for every step of the metagenomic projects and what are the usual difficulties and bottlenecks when you want to conduct metagenomic sequencing. Starting from the material collection, usually the best practice is, of course, as soon as you obtain the material of interest, to snap freeze it in liquid nitrogen and then store it at -80 degrees. because like this, we are sure that the integrity of the sample is not compromised. It is quite relatively straightforward to do in the hospital setting, for example. However, if samples come from the environment, then it is often challenging to ensure that they are directly not frozen or stored at the very low temperatures. So in this case, we have some recommendations. We anyway recommend to do as much cooling as possible, so to try to keep them at the lowest temperature available. There are also other options that can preserve mainly DNA. For example, one can lyophilize the samples or also use vacuum drying or fix samples. All this can be done, however, it has to be treated with caution because all of these techniques alter DNA and lead to fragmentation, which is usually really not good for the sequencing later on. And speaking about RNA, which is used in metatranscriptomics, it is always more difficult to preserve it than DNA, and we recommend a lot to use RNA preservants like RNA Shield or RNA Later to ensure that RNA comes in a good quality to our facility. One of the most important rules for the sample collection is that the freeze-thaw cycles have to be avoided. Of course, we cannot not to freeze-thaw our samples, however, they have to be kept at minimum, because this leads to DNA fragmentation, especially RNA fragmentation. And usually the fragments turn then so small that even though you have a lot of DNA in your sample, if you measure it, there is no usable DNA for us to sequence it. Going to the nucleic acid isolation step, next one after the material collection. Right now, nowadays we're really lucky because there are a lot of commercial kits available and many of them are very robust. So we usually use those. And what is also great that for many common metagenomic samples like stool or soil samples, there are tailored kits which include steps that are particularly important for exactly this type of sample. Something which you will hear a lot about if you want to collect metagenomic samples is the bead beating. That's the way to actually lyse the sample efficiently and to be sure that all the cell walls of bacteria are destroyed and we can actually access DNA. This is a very important step. Another step, which is also really important for metagenomic workflows, is the removal of inhibitors. Especially for soil samples, it's really relevant because humic acids that are present there inhibit any kind of enzymatic reactions, so we cannot process samples later on for the sequencing. And also, for example, gut samples or liver samples from human, they contain a lot of bile salts, which basically have the same effect. Another important point to keep in mind when speaking about the isolation of nucleic acid is the notion that host of the host organism will be also present in your sample. That is less relevant, of course, for environmental samples. However, for human patient sample, you will always have human DNA in your sample. And if for some samples, the amount of human DNA is quite low, for example, for urine or stool samples, it's 20%, it's quite high. Actually, usually it goes lower than that. So those samples we actually can sequence straight away. But for example, tissues like nasal swabs or some other bodily fluids, they usually contain a lot of epithelia. And that means that most of your sample actually is not only 95%, it sometimes goes all the way up to 99% of your sample is actually human DNA. And of course, then sequencing sample like this will be very expensive because one would need a lot of output to get relevant microbiome data out of it. So what do we recommend in this case? We recommend to introduce the host removal step in the isolation of during the nucleic acid isolation step. And there are different ways that one can do it. For example, during the sample collection, one can use centrifugation to remove human cells. This is the easiest way. There are also other options, like, for example, the differential lysis, because human cells are much easier to lyse than bacterial cells. So we can lyse human cell first, clean it, and then use the bacterial cells and get DNA from them. We can also use, for example, enrichment of non-methylated DNA, because usually bacterial DNA is not methylated, unlike human. So that also helps a lot. There is one last chance to remove host contamination is the bioinformatic removal. So after the sequencing is done, however, at that step, it might be actually too late to remove the host if most of your reads are from the host, then the information you get at the end is not really relevant. So we really recommend before commencing an isolation of nucleic acid for metagenomic research to get in touch with the sequencing facility and to ask what would be the best method of host depletion exactly for your sample, because at the end it will be crucial for the amount of data you get. So after the material collection and nucleic acid isolation step comes the library prep part. And here all the different methods I mentioned before, so amplicon sequencing, metagenomic shotgun, and also metatranscriptomics, here they have their own specialties. So I will start with the amplicon-seq. This is one of the most straightforward preps. And in this case, amplicon sequencing that I'm showing here is sequencing of 16S ribosomal gene of bacteria. And 16S ribosomal gene is quite a long gene, and it has several domains. It has variable regions, and it has 9 of them. We call them V1, V2, and so on. And it also has constant regions. And what is peculiar is that these variable regions are actually quite different from species to species. So by sequencing some of them, one can actually get information about the species composition in your sample. So how is it usually done? Usually we get the DNA from the sample, and then we go through several rounds of PCR. The first PCR is usually having primers homologous to one of the variable regions. So the most commonly used are V3, V4 region, and also, for example, V2 region or V5 are also quite common. We normally do the V3, V4. And then we run the first PCR, already having the partial adapter sequences on the primers. So this is kind of a half-ready sample for the sequencing. And after that, DNA is cleaned up, and we run a second PCR where we barcode the samples. So that's the part where we introduce the barcode to be able to demultiplex them later on. And after that, basically the samples get sequenced. Usually you don't need much of the output for this type of sequencing. So we'd use a smaller machine like MySeq. The important things to keep in mind when going for the 16S or amplicon sequencing is that it is only working for bacteria, because bacteria has 16S gene. Nowadays, there are also fungal markers established. So if you maybe heard of the ITS, this is it's not really gene, let's say it's the DNA sequence you can use for fungal species. However, one, for example, cannot cover archaea with this type of sequencing because they are so diverse that we do not have really a common gene that we can use. There are also markers, for example, for high eukaryotes, and one can use them for sampling of biocommunities as well in the environment. This sequencing does not require much output, and it's also quite cheap, and one can actually sequence quite some samples in one go. And what is also an important specialty of this sequencing, it requires pair and reads, and these reads have to be long, because the idea is that with the sequencing, you need to cover the whole fragment that was amplified to be able to provide reliable taxonomic information. So this is it for the amplicon sequencing. Let's now move to the library prep for shotgun metagenomics. So here we again start with the DNA of your sample, all the species that are present there, and after that, DNA undergoes fragmentation step. So there are different options available. One can use segmentation. It's one of the most popular ways. There is also sonication, quite popular, or enzymatic digest. And basically, we try to break DNA into fragments of a defined size. Usually, it's between 200 and 500 base pairs. After that, these fragments undergo ligation step, where the adapters are added for the sequencing, and that's also where the barcoding is happening for demultiplexing later on. And after that, the samples are amplified a bit to increase the amount of material that goes on the sequencer, and they're sequenced. So this workflow is quite straightforward, and it's also usually not that time-consuming. However, the important point here is that the sequencing output that one would need to analyze the data from the shotgun metagenomics is much, much higher than for the amplicon-seq. So here, for example, we're speaking about starting from half a gigabase, and this we call shallow metagenomics, because this is really kind of a pilot experiment that you can do to get the idea which species you have. And it can go even up to the 50 gigabases per sample if you really want to do deep metagenomics. For this approach, the host contamination that I mentioned before is a problem, because here all the DNA will be sequenced, so we do not enrich for the DNA of interest. So it's really important to remove the host before. However, for example, the reads that you need for this approach are much shorter, so it also simplifies a bit the sequencing procedure and also later on the handling. To summarize, the difference between the amplicon sequencing and the shotgun sequencing in terms of the preparation part, I made this table here. So speaking about the amount of nucleic acid you need, usually you don't need much for the 16S6 or for the amplicon, and you need much more for the shotgun metagenomics to be sure that all the species that you are interested in are actually present in the sample. As I mentioned before, amplicon-seq can be used nowadays mainly for bacteria and fungal species, and however, for shotgun, you can use basically for any species of interest. Much lower output is needed for amplicon-seq. So we're speaking about 20,000 to 50,000 reads per sample, which is really low. And transcribing this to shotgun, we usually measure it in gigabases. However, if you measure it in reads, it's from 50 million up to 300 million reads per sample. So you see the differences in several orders of magnitude. As I already also said, but I would like to underline it, host contamination is not that important for 16S because we anyway artificially enrich for microbial species. However, it is a big problem for the shotgun metagenomics. The library prep complexity is a bit higher for amplicon-seq, mainly because of the 2 PCR steps that I mentioned, because sometimes samples are coming from the really unusual environments and they contain inhibitors that even our kids cannot remove. So then the PCR step is complicated to get working. However, except of that, it's quite all right. And the library prep for shotgun metagenomics is really straightforward and quite easy. And what is also, it can be automated very well. Finally, the scale of the experiment supported. So usually amplicon-seq can be done in very large scales. So we're getting really often hundreds of samples from the labs, and we can actually sequence all of them at the same time. And for the shotgun metagenomics, because of the huge outputs needed, usually there are not that many samples that one can fit on a flow cell. And yes, last but not least, speaking of the price of both approaches, so amplicon-seq is not that expensive, and the shotgun metagenomics is of course getting more expensive if you need more output, if you need deeper metagenomics. Now I will give a short overview of the metatranscriptomic library prep. It is quite similar to the metagenomic. The only difference is that you start with the RNA. So of course, collecting the sample, isolating RNA. And what you get normally out of that is the bulk RNA of the sample. So there will be also ribosomal RNAs present in quite huge amounts. And this is usually not the RNAs of interest for researchers. So we go through the RNA enrichment step. So basically, there are ways to remove ribosomal RNA using special probes, and then to enrich the sample with the RNA of interest. There is also another way. One can use custom probes for exactly genes of interest. It is more complicated, but in principle, it is possible. For example, one can say, I would like to enrich my sample for the pathogens, and then we have special pathogen panel that would enrich only for those microbes and not for the others. So after the enrichment step, there is again, the fragmentation, same as for metagenomics. Then RNA goes through the cDNA synthesis, then we introduce barcoding step, and the library is ready for sequencing, and then it also goes on the sequencer. So why would one use metagenomics and metatranscriptomics and not, for example, just use metagenomics to discover new species? Well, metatranscriptomics brings new layers and new angles to the questions. For example, this is almost the only way to see what is the activity of the microbial community that you have, which genes are expressed, which genes are not expressed, and of course, genomic information will not give you this data. It is also often used in the healthcare research because one can check the microbial responses to certain therapies, or for example, to check the evolution of pathogenic genes and pathogenic responses in patients. Metatranscriptomics also comes with its own challenges. So first of all, it's indeed a very high output data, and if metagenomics already can go high, so metatranscriptomics in the output can go even higher, because to get the let's say sufficient resolution of the transcriptional profile, you need to sequence really deep. It is also really challenging to obtain good quality material because RNA is so unstable, and because there are also a lot of RNases usually present in the, let's say, environmental samples or also in the human samples. And what is complicated with metatranscriptomics is that the tools are not yet as good established, especially bioinformatic tools, are not established as well as the tools for, let's say, amplicon-seq or metagenomics. So often one has to develop their own tools to answer their research questions. So with that, I will close the library prep step. So basically, we now covered the material collection, the isolation of nucleic acids, and as well as the preparation of the samples for the sequencing. I also told you which kind of sequencing outputs we would usually use for different metagenomic approaches. And now I'll try to give you a brief introduction into the data analysis. So I must say that I'm not a bioinformatician myself, so I might not be able to answer all your questions about that, but at least I can give you an overview of what can be done in terms of the analysis. So first of all, it doesn't matter if you have your amplicon-seq data or your shotgun metagenomics data, they all go, so raw sequencing files all go through the same initial steps. Of course, they have to be demultiplexed, adapters have to be removed, and we also have to check the quality of the reads to be able to use the data of high quality for downstream analysis. However, after that, there are some differences appearing. So if we speak about the amplicon sequencing, so what usually comes after you have a high quality data, you try to cluster the 16S sequences you obtain. As you remember, basically you get a sequence of 16S gene from all your bacteria, and now you need to arrange them. So what usually is done bioinformatically is that you select a certain identity level that you want to have between the sequences for them to cluster together. For example, here it's 99%. You can also set it lower. And then the sequences are grouped together into so-called OTUs. Our bioinformaticians love this abbreviation. So that stands for operational taxonomic units. And it's sometimes a bit difficult to understand for the scientist what it is, but let's say it's kind of arrangement of the most similar sequences together. One cannot say that this is arrangement by species, because still it can be broader than that. Usually it's a level of genus. However, that's an artificial way to group similar sequences. And then these OTUs are usually compared, so they're mapped to the existing database. So they're really well curated databases now for the 16S sequencing with really good data, because basically this method has been done for many, many years. And after the annotation of these OTUs with the help of the database, you can get a list of species that are present. So usually not species, sorry, the list of genus that are present in your samples. 16S unfortunately cannot go all the way down to the species. However, genus is often sufficient. And with this list, one can do then further comparisons. For example, the most common comparisons include alpha diversity, which is assessing the diversity of species within the sample. So for example, if you have a patient gut microbiome sample, you can check which species are present and in which amounts. Or the next level would be the beta diversity. And this is a comparison of composition between different samples, for example, between patients or before and after treatment. Speaking about shotgun metagenomics, it goes through a bit different way. So after the quality checks and the read trimming, one would go through the de novo assembly of the sequences. So important point as well, you can also choose to, for example, remove host reads at this point, as I said. So then you're left only with your microbiome reads. And then there is a computational process of trying to kind of stitch the sequences you obtain together into artificial genomes. And these genomes are later on again bind together according to their similarities. And after that, they are taxonomically classified. So basically, you try then to map them to existing database and annotate the organisms and the genes. So this approach can go all the way down to species because you get much more information from the sequences you have. So they cover not only obviously 16S gene, but also other genes. However, I must say that for species resolution, one has to really sequence deeply. Normally, people also go down to the genus resolution to get the taxonomic data. To give you a bit more information about what both methods can do in terms of bioinformatics and in terms of the data you can get out. So here's again a comparison of amplicon-seq with the shotgun. And again, as I mentioned before, amplicon-seq is mainly used for bacterial fungal species, when shotgun can basically detect any kind of species. The analysis pipelines for amplicon-seq are very developed and very well curated. So for example, also non-bioinformaticians like me can also run a pipeline. The metagenomics pipelines are improving a lot. So they're also getting very well curated, and they're also quite easy to understand. However, one still needs a bit more bioinformatic knowledge to be able to do this kind of analysis. Speaking about computing power, usually amplicon-seq does not require that much computing power, and the analysis goes really fast. While metagenomics, shotgun metagenomics requires much more, and it also can take some time. Taxonomic resolution is, yes, genus level for 16S, species level for shotgun, but again, it has to be really deep sequencing. And then these are the types of assessments you can do. So for example, comparison of what I mentioned before, comparison of the species, quantitative comparison of the species within the sample is possible with both methods. Taxonomic comparisons as well, of course. But for example, new genes and new species discovery is not possible, of course, with the amplicon-seq, because you get information only from one gene. On the other hand, metagenomics and metatranscriptomics will give you that data. And also, if one would want to analyze the active pathways, biosynthetic pathways, or any other actively transcribed genes, then it would be only metatranscriptomics that can give you the answer to that. All right. With that, I will, okay. So yes, coming back again to the overview of bioinformatic analysis. So there is also a, let's say, in-between way between the arranging the OTUs and also the de novo assembly, and it's also used often to get a quick look at the data that you get. And this is the mapping according to the lowest common ancestor. And one of the most used tools for it is Kraken. So most probably you heard about it. And right now that's the newer version, so it's called Kraken 2. This is a good way to get a quick look at the data and to, let's say, look at what might be interesting and what one should focus on. So how Kraken works, it basically breaks the sequences you get into smaller chunks. So they are called k-mers. And then it kind of gets shifts the frame of the cutting and then cuts it again. So you basically, from one sequence, you get many k-mers that are partially overlapping. And after that, these k-mers are mapped to the database. And what is happening there is that basically Kraken is checking what is the lowest common ancestor. So what is the kind of lowest level it can go to and map most of the sequences. And some of the sequences will be already sieved out quite early just because they are these small chunks, it cannot really assign somewhere, but then it would make a decision on the tree, since most of the sequences will, let's say, map to a certain bigger group of species, then it will go down further, and then, for example, it will define, let's say, genus level over here. Usually Kraken is also not enough to determine species, however, determining until the genus level is quite easy. The tool is really popular, however, I must say that using only Kraken for taxonomic analysis is not really a good way to go, because it has to be verified also by other methods. However, it's really easy to use, and it also can be used by non-bioinformaticians just with a bit of extra knowledge. Now, the final part of the bioinformatic analysis of metagenomics is the ways to visualize data. Of course, this is also really important because that's how they will be presented to the scientific community. And there are different ways one can do that. One of the most popular ones is the histogram with the microbial profiles. So for example, that's how the typical 16S data would look. And basically, one would plot different genera of bacteria on the histogram correspondingly to their amount in percentage. So here on the x-axis, there are patients, and basically you can compare not only the amount of different genera in one patient, but also the amount of different bacterial composition within different patients. Another really popular and really impressive way to visualize the data is the corona plots. And corona plots are basically, yes, it's in a way pie charts with the distribution between different species. And they are also giving a good idea about if you want to have a very visual idea about how many species are represented in which mounts, that's a great way to go. The only problem is that corona plots can be generated only for one sample per one plot, so then it gets bulky if you want to compare several samples. Finally, this is something that is mainly done with metagenomic analysis. This is visual gene annotation. And basically what you see here, these are different contigs assembled from the sequencing data, and one can annotate the genes and try to tell it to which kind of gene class they belong, whether they are responsible for DNA replication or maybe for the structural proteins or for something else. So it also gives this is now a phage genome, so it also gives you a great idea about what are the main elements in the genome of your species of interest. However, I must say that this kind of visualization, it's a complex version of visualization of metagenomic data. All right, with that, I would like to round up with the bioinformatic analysis. Let me speak briefly about the applications of metagenomics, and they're actually vast. That's why I don't want to spend too much time on that. Of course, the applications in healthcare are the most obvious and those that we hear about all the time, because yes, it can be used in diagnostics. It can be also used in search for novel therapies against infections, also biomarker development and other applications. It is also, metagenomics is also really widely used among environmental scientists because that's a great way to assess the quality of water or soil, and also actually still to discover new species of microbes that are out there. Agriculture is actually one of the biggest contributors in the metagenomic services. So I did a bit of search for this talk, and 2/3 of the samples that are submitted to the commercial sequencing facilities are actually from the agricultural sector, mainly interested in soil health and also in health of the cattle. The biotechnology also uses metagenomics a lot, and one of the most known uses the wastewater management. So one would also need to sample the health of the microbes there. And even the areas where one would not really think about metagenomics, like forensics, they're also finding it a very useful tool, for example, for soil sampling and comparison it to the possible places of incidence. And metatranscriptomics basically can be used as a part of metagenomics, can be used also in all those approaches, but this is a method that adds yet another level of data you can read. It basically gives you the snapshot of the activity of the microbial community from the sample you're interested in. So what are the kind of next steps in metagenomic research and what we are trying to develop right now? So now metagenomics of long-read sequencing is coming on the stage. And I think you already heard a talk about long-read sequencing within the GHGA seminars. So I will not really explain much here. I'll just mention that there are two main platforms we're using nowadays, the PacBio platform with this random circle PCR, and also the nanopore platform with the pores and the current monitoring for sequencing. And both of these platforms are used actively for metagenomic sequencing. And they offer quite some advantages because the taxonomic profiling is getting much better because we also get longer sequences and it's easier to map and to assemble the genomes. And yes, as I said, the genome assembly takes less effort and less computational effort as well with longer reads. And finally, it also gives an information about complete genes and often gene clusters or even huge chunks of genomes, which is also really useful for example, prediction of biosynthetic pathways in microbes. And another direction where metagenomics is developing now is the multi-omics approaches, basically when there is multiple levels of information obtained from one sample. And I would say, so now we can obtain genomic and transcriptomic information from one sample and even proteomic snapshot. This is not, it's still quite challenging technically, however, it can be done. However, now the next level is also to, at the same time, to monitor the metabolomic profile. And this gets quite tricky with the microbiome, because it's just so many things one has to isolate, and often samples are just simply not having enough input material for that. However, the applications of these multi-omic methods are, of course, incredible, because they will really revolutionize healthcare and also personalized medicine when they become a bit more widely used. So this was the quick introduction into metagenomics, a really broad field of sequencing, and also really filled with a huge potential. And on that, I would like to finish my talk and to thank you all for listening. And also, please refer to us and also to NGS-CN network if you have a metagenomic project in mind, and we will help you as much as we can. Thank you.